Very quick overview and update on AdAlta's activities. Everyone knows I'm the CEO of AdAlta. My background's business development. I've got a science background and law and have been the founding CEO of AdAlta for the last 10.5 years. Worked in AdAlta and seen it through different stages, getting to a very, very exciting time in the company, looking to get AD-114 into the clinic later this year. So I guess as we've heard today, we've got a very, very exciting platform technology where we identified AD-114 from. Our initial focus with AD-114 is in the treatment of fibrosis. We've talked about the significant unmet medical need in this space. There's been 2 approved drugs, pirfenidone and nintedanib. They have massive side effects. They have paved the way for a drug like AD-114 in terms of getting into the clinic, and the bar is low for other drugs, and the opportunity there exists there for new drugs to come through and improve on what they've done to date. We IPO'd in August 2016, raising funding to take AD-114 into phase 1. That's our focus. We got orphan drug status for AD-114 early last year. And we've spoken about the commercial opportunity in the fibrosis space. I think a few people have mentioned fibrosis is a hot area. We see a lot of deals happen in this stage at the end of phase 1, which is very, very early for a clinical product. As you've seen today, I think we've got an experienced team, amazing scientific advisors, an experienced board in terms of taking products through the clinic, raising capital and exiting also from those assets. So our financial position, I think somebody just told me we're 31.5 cents since this morning. Increased. Sorry? 30 more cents. Yeah, that's right. Oh, sorry, Stuart. Yeah. UR Capital being our main shareholder, and Bianca, who you heard earlier from Platinum Asset Management as well. City Castle, I think Leon might have just left. And then La Trobe University, and then national nominees being PPM. So we have a very, rich shareholder register in terms of both institutional investors and high net worths in this space. So we've heard a bit about the iBody platform. Essentially where it came from, so I think I heard somebody mention shark earlier. What we did was we took the shape or the structure of that shark. So the nanobody or the camel antibody and the shark single domain are the only 2 antibodies that have evolved to have a single domain antibody. We found the structure of that, and from identifying the structure, found a human protein that was essentially equivalent in shape. And so then we've taken that shape, the red bottom part, and as Mick mentioned, then incorporated different loops at the top of that molecule. And those loops provide the diversity in the library that we have. So we have 20 billion different iBodies that have different amino acids in those 2 loops at the top of the molecule. So we've spoken about that long loop and how it's a competitive advantage of the iBody when we're looking at GPCRs and ion channels and able to bind to, like small molecules do, into cavities and new epitopes. These iBodies are also extremely stable like a small molecule and provide us the potential to look at alternative routes of administration. So the iBody has some properties of small molecules. We also, as a single domain antibody-like molecule, also have properties of the conventional antibody. So we heard Mick today talk about the high specificity of AD-114 and also the selectivity that it has. So we would expect, because of that high specificity and sensitivity, that we would get less off-target effects, which you do see with small molecule drugs. So really the AdAlta iBody is combining the advantages of both small molecules and antibodies to create this new platform technology for drug discovery. Fibrosis is a significant unmet medical need. 45 to 50% of all deaths are actually have a fibrotic component. We see it in the lung with the disease Idiopathic Pulmonary Fibrosis that we're focused on, but we also see it in conditions in the eye, so wet AMD or age-related macular degeneration, in liver cirrhosis, NASH, kidney fibrosis with Carol Pollack talking this morning about the data with AD-114. And in all of these areas we see that there's either no drugs or drugs that aren't addressing this huge unmet medical need. And I think to Derek's question earlier, we see with CXCR4 a significant upregulation of this target in disease. And we're looking at human tissues here. So we look at the normal tissue and we see no expression of CXCR4. In the disease tissue, CXCR4 is just basically lit up. And we see this in the kidney, we see it in the lung, and it really shows that this product is specific for the disease. The bottom two tissue pictures are really showing that in idiopathic pulmonary fibrosis, we've looked at the whole lung, and the areas that CXCR4 is present is really the active edge or the leading edge of fibrosis. So initial data that we had going to IPO was really in the bleomycin mouse model. So this is a mouse model of idiopathic pulmonary fibrosis. So on the left you see the normal tissue, the normal lung tissue. In the middle, the IPF disease tissue where the mouse is being given bleomycin. And on the far right you see the disease tissue given with AD-114 for 21 days. So you see a significant reduction in the blue protein, which is collagen. We also see a significant reduction in the inflammatory cells. So we measure this by a score called Ashcroft score, and we see that significantly reduced. We see a negative I-body having no impact. I think Mick went through this slide in detail. One of the key differences between AdAlta's AD-114 and other CXCR4 drugs is that we are not mobilising stem cells. And as he mentioned, when Corey Hogebone completed the evaluation of AD-114 with human IPF tissues, we saw a significantly different profile to the existing treatments, so nintedanib and pirfenidone, and to the CXCR4 drug Mozobil. So AD-114 has a very specific profile for treating IPF. It has an effect on IPF fibroblasts and does not have an effect on those normal cells because of the expression of CXCR4 in those tissues. A very interesting small molecule PET imaging agent has been used by a German group to look at the effects, the expression of CXCR4, but also the effects of pirfenidone. So the top two pictures in this slide are of a patient that has very low CXCR4 expression, and then the far right side has a picture at 6 weeks after they've been treated with pirfenidone. And you see that CXCR4, when they've got low CXCR4 expression, they're responding to pirfenidone. The bottom two pictures are of a patient that has very, very high CXCR4 expression, and that patient did not respond to the existing treatments. So this group has been looking at CXCR4 and saying that CXCR4 imaging may have a role in monitoring disease, but also may predict a response to the treatment with pirfenidone. So using a small molecule PET imaging agent, and I think John might have mentioned this, as a PK/PD marker as well in our clinical trials. This is potential to get an early indicator of how we're looking at the dosing for CXCR4 and AD-114 in the clinic. We've spoken about the fibrosis area. These are a list of the deals in IPF since 2011. And what we see is the average upfront payment is about $100 million. The deal size is between 400 and 500 million. And these deals happen at phase 1. But more recently we've seen 2 companies read out phase 2 studies. The bottom one being Galapagos had a phase 2A study where they had 17 IPF patients and saw a market increase in their company of about $500 million. And Fibrogen, a phase 2B study with 100 patients. And I think this really talks to what the panel were saying about progressing that drug to a clinical proof of concept and creating additional value in the company. What we've shown over the last 12 months is really that it's not just one model that we've seen AD-114 have activity in. We've seen it across several different animal models. We've seen it across several different types of human tissue. We've seen it in the lung, we've seen it in the eye, the liver, skin and kidney. We've also shown in our first 3 non-human primate studies that this drug is extremely safe and has no off-target effects. So some of the data in the eye, we see compared to Eylea, it's exactly the same in terms of measuring the lesion size, and also very similar in terms of fibrosis. But Eylea only works with about 70% of patients, and after 2 or 3 years, 50% of people start to decline and go blind. So there's a real need for a different mechanism of action in this space. In the liver, again, we also see AD-114 working. So on the far left here, we've got a normal liver, the middle is a diseased liver, and on the far right, you see also in a therapeutic mode, AD-114 reducing those fibrotic effects. And we see these including serum ALT, an NAFLD activity score, and ballooning being significantly reduced. So today we've really spoken about, well, AdAlta and the platform sitting behind AD-114. And it really provides us an opportunity to do partnerships as well as in-house drug development. We've got 20 billion compounds in this library, and really doing both of these provide us an opportunity to bring in revenue, but also to grow the shareholder value. With this platform technology and AD-114, we see really 3 main areas for the company to build value for shareholders. So the first being AD-114 with a deal in fibrosis. Next generation antibody companies, we've spoken about Ablinks being acquired. I think Bianca mentioned Pieris and Argenx. These companies are active doing collaborations as next generation antibody companies with significant upfront payments and development milestones. In the GPCR space, Robert's example with Receptos, Ablinks again, as well as Heptares, which Anthony mentioned. So 2017 was a big year for us. We kicked off with our manufacturing with Fuji. We licensed the pacylation technology for the half-life extension. We've received a number of grants, including with the Alfred here, but with Carol Pollock. We've also presented at a number of conferences, both myself and Mick, as well as a number of partnering conferences, talking to potential partners about the platform and AD-114. We've had Robert joined us in late 2016. Dallas also joined us late last year, 2017. So we've really strengthened the board, our scientific advisory board, as well as our management team. AdAlta received orphan drug designation in the US for AD-114, a very important milestone and validation of the technology and the lead candidate. And I guess cross-beta is an example of a product that we've licensed out. So a shark antibody identified to A-beta oligomers, and we've licensed that to a company called cross-beta. We'll receive milestones and development milestones and royalties on anything that they develop in that space. And like last year, this symposium's really aimed at bringing investors, analysts together to talk about fibrosis and AdAlta's platform technology. So we've really seen over the last 12 months a real interest in what we're doing. So both myself and Mick have been invited to speak at several conferences. Myself at the Idiopathic Pulmonary Fibrosis conference, and Mick at a general fibrosis conference, as well as at ARVO, the eye conference. We continue to talk to media and drive the interest around the technology, and the BioShares guys here also mentioned us as one of the top 4 potential acquisitions for 2018-19. So we'll see.
Well, do they know that you don't?
I don't know.
We just were speculating.
So key milestones. Our first half of this year, toxicology studies, our 4-week tox study. Second half of this year, getting into phase 1. So really, but sitting behind that, publishing the data and talking to partners about both AD-114 and the platform. So in terms of expected news flow, we've had a great year last year, keeping investors up to date on what we've been doing, and looking to 2018, as I mentioned. We'll provide an update on manufacturing shortly, the update on the 4-week tox, and publication of our lung data. All of these things will come in the first half of this year, and the second half will all be around our phase 1, getting into the clinic. I think as you've seen today, we've got a great board joined by Robert Peach, James and Liddy from UR are also here. John Chiplin couldn't be with us, nor could Paul, but we've got an amazing board with strengths in both capital raising, exiting from companies and drug development. We've heard from a few of our scientific advisors, Mick being our Chief Scientific Officer, Brian and John, and a few comments also from Steve. I think you would agree with me that these guys have an amazing experience in terms of drug development, like nothing I've ever seen. And I think Stuart's going to tally the drugs that they've developed. So in terms of an AdAlta summary, 2018 is a very big year for us. We're looking forward to it, getting into the clinic. Our initial focus is on idiopathic pulmonary fibrosis, but we do have an underlying platform that creates significant value for the company. We've got an early commercialisation opportunity with AD-114 and a great team to get us to that point. So thank you for coming today. It's been a great opportunity to talk to you about it. Thanks. Yeah, go for it.
Can you go back to the slide where you list the 6 different tissues? Go forward there. Can you tell me what the duration is of a proof of concept trial in each of those areas, noting that somebody said for 2 years, I think, for NASH, or is that phase 3?
I think that...
So with the IPF, you're looking at, what, 12 weeks?
So the Galapagos trial had a 12-week readout, but they're traditionally all 12 months. So lung IPF, the phase 2B was a 12-month study. Forced vital capacity is the standard readout in that space. They are doing secondary readouts like imaging, so HRCT scanning in those areas. And they're trying, and I guess that's Galapagos use that to get a significant readout in their phase 2A study with 17 patients and a 12-week time period. But in IPF, a traditional phase 2 study is a minimum of 12 months looking at forced vital capacity. In the eye, again, 12-month readouts. You're looking at, you guys will be familiar with OPSEA. Yeah. You could do earlier readouts as well. You can do 1 month recovery periods. I think that you can look at shorter time periods as well in terms of looking at the line reading, as well as looking for, I think in this instance, looking for subretinal hyperreflective material, so SHREM, and doing OCT scanning. So I think that, we're looking at the clinical plan in these instances and saying, well, could we get a readout? What are the patient numbers that you need? I think there'd be significantly larger trials than an IPF trial, and maybe similar readout times. I think, like we said, liver, I think they're 12-month minimum trials. The kidney, I guess James and Lydia would be more familiar with this with dimerics, but it's shorter readout times, I guess, for kidney disease as well. The skin and heart, I'm not sure. We haven't looked at those in great detail.
Okay. With the phase 1 trial, was that second half or
yes,
first half?
Second half.
Second half. And can you just explain how that trial's going to run?
Yep, so there'll be a SAD, single ascending dose. We're looking at 6 groups of single ascending doses. Then we'll move into the multi-ascending dose, so MAD study. And we'll also look at trying adding in some IPF patients in some of these to get some earlier readouts, PK/PD, and PK readouts.
So that's all the
all male healthy volunteers.
So that'll be 2 in the first...
Not New Zealanders.
Will that be 2 and then you wait a month? Will that be 2 and then you wait a month?
No, we won't wait a month. We'll have a washout period in between, but it will not be a month. No, it'll be a lot shorter than that. Yeah.
Sam, sorry, just on the IPS patients, sorry you plan to put in that trial? During the washout on those patients, or are you still...
No, so we're still looking at that design with the IPF patients. I mean, the single ascending dose and the multi-ascending dose with healthy volunteers is actually much more straightforward.
And what the timelines as far as finishing now that study?
Yeah, so we're putting those together at the moment. I mean, so like we have to look at washout periods. We'll have more data when we've finished our 4-week tox study and the PK/PD assays, but we want to run this pretty quickly in terms of the single ascending dose study and get some readouts as soon as we can. Yeah. So it'll be 6 ascending doses.
Sub-Q?
Yes.
And if I can back up, sorry, if I may, manufacturing, GMP material, how's that going?
Yep, so we'll give an update to everybody at the end of February. going well.
And then in the talks, there's the 4-week talks left,
Yep.
and that's started now, it's currently going to start.
The materials we're making, it'll start when those materials are ready.
Is it decided what to do as far as partnering with the program? Obviously, we've heard the panel about, do you say or do you go? What's the best way to do it? Obviously, it's an ongoing discussion amongst
Yep.
the panel, the scientific review board.
Yeah.
What's your thoughts as far as, do you let this asset go? Someone comes to your
Yep.
door, Sam, and I have X amount to give you. Do you
Yep.
say, okay?
I mean, I think it always depends on the deal that you've
Sure.
and the opportunity. I mean, I think as a board we are focused on taking AD-114 through a phase 1 study. We will be in a position to say whether we're partnering that at the end of phase 1 if we've got an opportunity, I think. But we've also got to look at what a phase 2 proof of concept will look like and creating value for that as well. So I think for us as a board, it'll depend on what opportunities are available. We'll look to do a partner deal as soon as we possibly can with AD-114, if that means it's the best deal for shareholders. If not, we'll look to raise the funding and create more value. But I think from a partnering point of view, again, I think a number of the scientific advisors and board members have said, we are strongly focused on AD-114, but we're busy over there building a pipeline of iBodies, looking at where these advantages of the iBody platform can be with ion channels and GPCRs, and try that good data package, which I think these guys have mentioned, is really key for me to go out and partner. Again, additional resources. I think it comes back to David's point. It's all about we need additional resources to take as many of these things through as we can.
Yeah, and that phase 1 design, is it a phase 1/2? Is it rolling into a phase 2? Or is
At
it
this stage, we've got funding for phase 1. At this point in time, we've got funding for phase 1.
What's the competitive landscape like for partnering? I mean, do you
Yep.
have an excess of potential partners versus companies like ourselves that are chasing partnerships? I mean, how does that play?
Yeah, so I mean, I think the pharma industry is, very active in looking at companies like AdAlta in terms of partnering. They know exactly what they want from that point of view. I've met with a number of them and they've told us exactly what they want to see. I think also that in terms of the pipeline of IPF products, there'd probably be there's no products in phase 3. There's the 2 approved products. Actually, I think that changed this week. I tell a lie. There was 1 phase 2 product that went to phase 3, Promedic, and that was a FDA fast track product because the phase 2 data at 12 weeks looked great. So you've got a product moving from a phase 2 very, very small trial to phase 3 really rapidly. You've got a pipeline of about 10 products in phase 2, which is very, very small. You've got 2 readouts, one from Galapagos and one from Fibrinogen, and we've looked at that data and think that what we've got is competitive. You've got probably another 10 products in phase 1. So the pipeline of IPF drugs is nowhere near a cancer pipeline or a NASH pipeline, and we think we can compete in this space. We've got a different product, different mechanism of action, and we just need to get into the clinic and show that it works.